Okay, so chapter one is on just introducing you to genetics, and it will cover things such as a lot of history that led us to the point of where we are in genetics, how it has built the foundation for what we know about Mendelian principles or transmission genetics, even biotechnology, genomics, proteomics, bioinformatics, and how we use genetics in modern day. For your own learning, these are where the learning objectives can be found for each of the sections within the chapter. So I like to open up each chapter with a case study or some type of relevance to what we know today. And one of the important factors I think that we should talk about when you talk about genetics is CRISPR. CRISPR is a technique that stands for CRISPR stands for clustered regularly interspersed short palindromic repeats. That's a fancy way of saying it's a technique that is used to modify the DNA of a said organism of interest, where you can actually cut out, or like scissors, a portion of the DNA that you no longer want to be there. So you can modify the genetic information in simplistic terms. Now, this technique, CRISPR, was done on Chinese twin babies who have been named Lulu and Nana. And the scientist that carried out this process is He Jiankui. These twins were born in China in 2018, and their genome was altered by the scientists using CRISPR to target HIV resistance. And this has sparked much debate. One, this was a lot of questions around the ethics behind what he did, and was it okay for him to, in essence, edit the CCR5 gene, and what are the downstream effects? Now, the truth is, they don't know the downstream effects, and what may happen to them. And this is one of the issues why using CRISPR in certain levels is um, illegal or is frowned upon. And this is one of the issues that many have had with him. The scientist was actually sentenced to time in prison. I'm not sure if he's gotten out yet, but he was sentenced to time in prison for his unethical behavior and his inability to follow regulatory laws and policies. Now, while CRISPR may sound to some as crossing ethical lines, one thing that we do know is it is an editing tool that has great benefits. It was initially discovered as a seek and destroy mechanism in bacteria, and now we're able to use it to fight and develop treatment for various diseases that affect humans as well as agriculture. For example, it's used for treatment of sickle cell disease and beta thalassemia, which are both blood disorders targeting the beta globin gene, but we also see the use of CRISPR in agriculture, such as disease resistance, drought resistance, and helping to improve the nutritional value in various vegetation. So CRISPR is not always bad. And I want to point that out because we're going to start talking about how history has built itself over time. And it led us to this point, but understanding the etiology of why these biotech techniques have started and existed have not been for unethical purposes, but more so for the betterment of humankind. So let's talk about some of that rich history. Sometimes when we talk about genetics, it can be daunting because people feel like it is something that is so far from their reach in terms of grasping the concepts. But I want you to know that humans have been using genetics for thousands of years. If you look back 10 to 12,000 years ago, it's been a form of genetics has been done in the domestication of plants and animals. Whether you're talking about the first domesticated wheat, peas, lentil, or various animals like sheep, goats, you also had instances where Aristotle and Hippocrates uh, posed the concept of the active humors, whether they were healthy or diseased. And um, they ultimately, Aristotle thought that these active humors in various parts of the body served as the barriers for hereditary traits, and that they were drawn from various parts of the male body to the semen and then passed on to offspring. These humors or seeds could be healthy or diseased, and with these diseased humors accounting for the appearance of newborns with congenital disorders or deformities. They also believed that these humors could be altered in individuals and then passed on to their offspring, and it is believed that this passing on of these traits or humors to offspring explain how newborns could inherit traits from their parents and have an acquired resistance or acquired inheritance or response to their environment. Now, what was further interesting is that Aristotle extended Hippocrates' thinking and proposed that the male semen could contain some type of heat or vital heat, and this was had the capacity, to produce offspring of the same form. And so ultimately, this vital heat from the semen, would shape or cook the female blood to give rise to the offspring that you see. Sounds kind of far-fetched, right? But they were in the beginning stages of not fully understanding certain concepts, but this was their theoretical proposals that they put out there. But I want you to also consider that there are certain concepts that predate certain eras. So, for example, even in ancient Jewish writings, in the what we call the Talmud, there was an understanding of hemophilia, even though they did not understand that's what it was. They didn't call it hemophilia because they hadn't been termed hemophilia at that time, but they proposed that concept. And what they ultimately stated was that if a female or woman had a child, had two sons, and the sons uh, bled after circumcision, that she should not circumcise any of her other children that she would give birth to, and to also inform her sister that she herself should not circumcise her boys. So this, while they didn't understand exactly what they were talking about, right, exactly the terminology, what this advice accurately stated was that it corresponds to X-linked pattern of inheritance of hemophilia, and it suggests that it was the female who had a higher chance or probability of giving that trait to their sons, and that is what you see in their terminology in the Talmud. You also have the concept of uh, theories of inheritance from ancient Greeks, and pangenesis, which was an early concept of heredity, proposed that particles carry genetic information from different parts of the body to the reproductive organs. While it was incorrect, it highlighted the inquiry of human and genetics. And so ultimately, what you're seeing is these different pieces of history hinting to or alluding to different components of science of genetics that we know have evolved over time. Also, think about the theory of of inheritance of acquired traits, which is no longer accepted, but it was proposed and somewhat popular throughout the 20th century. You have Darwin, who, the dawn, of modern biology. You have the theory of epigenesis, which suggests that there are structures such as body organs, and they're not initially present in the embryo, but are formed later in adulthood, which sounds more reasonable than the next, which is the theory of preformation. And the theory of preformation says that you have this homunculus, or this miniature adult that is within the fertilized egg, and it's already fully formed. It just enlarges over time. Now that sounds very far-fetched and almost like a Honey, I Shrunk the Kids type of scenario. But this was, again, another proposed theory that existed. Now, I mentioned in the previous slide the inheritance of acquired characteristics or traits, which was proposed by Jean-Baptiste Lamarck. And he proposed that traits are acquired in a person's lifetime and they're passed on to offspring. Now, you'll see here in the right side of the screen that there's these images or these theories images to explain what Lamarck's theory was. And it started from ancestral drafts having to stretch their necks to reach the foliage in the trees. And with each generation, their offspring inherited that stretched neck to the point where now they have these long necks that allow them to reach foliage in the tree, just through repeatedly inheritance of stretched necks. Now, we know this is not true, because if this theory of inheritance of acquired traits were true, then any traits that your parents have, you should acquire them as well. For example, my father's ability to play the bass, he's great at it. I am not. According to inheritance of acquired traits, if my father gained the trait or the ability to play this guitar or bass, then I should too. But that is not the case. So there's suggests a flaw in this concept. You also have the Schleiden and Schwann's cell theory, which suggests that all organisms are composed of basic structural units, and those structural units are called cells. Now, what is interesting about this is while this was proposed back in the early 1800s, a form of this we still accept, right? We know that organisms are composed of cells. And so this just had to be a theory that was built off of. Louis Pasteur, with his theory of disproved spontaneous generation, and that ultimately he conclusively disproved the theory of spontaneous generation in the 1860s with his famous swan neck flask experiments, which demonstrated that microorganisms come from pre-existing ones, i.e. biogenesis, rather than arising from non-living matter. And how Pasteur did this was he showed that when sterile broth was exposed to air through a curved neck that trapped dust and microbes, no life grew. No life was seen or formed. But they only found that when the neck was broken, allowing dust to enter in and microbial growth occurred. And then we have Charles Darwin. Now, Charles Darwin published his groundbreaking theory of evolution by natural selection in the Origin of Species, which was in 1859. And this was a series of observations from his HMS Beagle voyage that took place from about 1831 to 1836. And he introduced concepts like the descent with modifications, where ultimately he proposed that species gradually change over generations due to traits that allow them to survive or were advantageous or beneficial to them. And his work that he did over time really was not accepted until later on. But his work did give rise to the concept of natural selection, which is the actual mechanism for evolutionary change. The actual theory of evolution was independently proposed by Alfred Russel Wallace. So now, how do we get to this whole concept of genetics, modern day genetics, or even the whole concept of DNA and traits being passed on? We credit that to the father of genetics, which is named Gregor Mendel. Gregor Mendel was a monk and he's specifically an Augustinian monk who conducted a long body of work. Took him decades to do so, but he used a series of experiments that used pea plants as his model organism. And he took pea plants, but he applied a quantitative, meaning mathematical approach to analyzing his data that he collected. And he ultimately proposed that traits are passed on from one generation to the next, and that this transmission of genetic information went from parent to offspring. His work was so profound that it still resonates today, and it serves as the foundation for genetics and science as we know it. In order to fully understand, the whole concept of transmission of genetics, you have to have a understanding of mitosis and meiosis. Now, when you look at a lot of the work that was done over the past century or so, within the 19th century, it described, a lot of research described chromosome behavior and how they work in ultimately two forms: cell division, which is mitosis and meiosis. Now, in mitosis, chromosomes are copied and distributed, and the resulting or the product of mitosis is two daughter cells that are diploid and identical. Meiosis, on the other hand, you have chromosomes that are copied and distributed, but it results in the formation of haploid gametes, i.e., sex cells. Okay, so this is important because this helps us understand, one, the differences that cells go through based off of their cell type, but also their fate. And so it was early in the 20th century that Walter Sutton and Theodore Boveri independently noted that the behavior of chromosomes during meiosis is identical to the behavior of genes during gamete formation that was described by Mendel. And so this gave rise to a clearer understanding of the difference between these cell types, but also actually what is happening within a cell. And this information literally led to the formation of the chromosomal theory of inheritance, which states that inherited traits are controlled by genes residing on chromosomes faithfully transmitted through gametes, maintaining genetic continuity from generation to generation. Another way of saying that is that due to this faithful or consistent passing on or division of traits, that there is a level of consistency that occurs. Okay. So when we say diploid or 2N, we're referring to organisms that have two copies of their chromosomes. Most eukaryotes have a characteristic of number of chromosomes, and we call that diploid or 2N. Now, when you hear that concept of diploid or 2N, think of eukaryotic cells that have, or human. In this picture here, you have a human male karyotype, but there are 23 pairs of chromosomes. So it'll be 2 times 23. That's where the N comes from. Okay. But ultimately, chromosomes exist in pairs when they are diploid, and they're called homologous chromosomes. For example, in this diagram here, you see that in the male human karyotype, which is a map of the chromosomes, that there are 2 copies of chromosome 1, there are 2 copies of chromosome 2, and 2 copies of chromosome 3. And while there may be small differences that you can't see with the naked eye, ultimately their morphology is very similar or the same. The only major difference that you see in the morphology between these chromosomes is X and Y, which are your sex chromosomes. Now, as I mentioned, the chromosomal theory of inheritance that was proposed by Sutton and Boveri proposed that genes are carried on chromosomes. Now, what is a gene? A gene is a portion of DNA, which is the molecule, right, or the vehicle that carries genetic information. And this theory of inheritance was independently proposed. It was built off of work that was prior done that we've talked about, but it was proposed independently. And it suggests also that inherited traits are controlled by genes that reside on chromosomes faithfully. Now, when we talk about different traits and how they're passed on, I want you to think about gametes, because it's important to understand when we're talking especially about diploid organisms that these gametes are responsible for passing on genes through inheritance, and it is the genes that provide continuity or consistency from one generation to the next, and that further helps explain Mendel's law that will be discussed in chapter 2. Okay, so this diagram here shows you a chromosome in flies, and on this chromosome, there are various genes that lie on this chromosome that are responsible for various traits, such as wing size, the color of eyes, the color of the body, the shape of the eyes. These are examples of what chromosomes carry. So, what are alleles? We talked about genes. Genes are a portion of DNA that are responsible for heredity or carry information for heredity. So, what is alleles? Okay. Now, part of our understanding of this came from scientists beginning to use fruit flies or Drosophila melanogaster as their model system to understand the differences between different traits. And from that, they were able to understand more about alleles. What is an allele? It's an alternate form of a gene. Well, how do we get alternate forms of a gene? Well, mutations, right? When you hear the term mutations, oftentimes it has a negative connotation to it, but actually mutations have a benefit because they help provide genetic variation, and they're actually considered to be the source of genetic variation. And so when alleles or genes produce or inherit mutations over time, they form an alternate form of themselves, which we term an allele. And these alleles together form a genotype, which determine a given trait for an organism, whether we're talking about fruit flies and whether they have red eyes or white eyes, or the shape of the eyes, which we would consider the phenotype. The genotype is the set of alleles that give instruction, and the phenotype is the expression of that genotype. So what you can see with your eye, what is observable, right, would be considered the phenotype. So genotype determines phenotype, and phenotype is more observable than genotype. And that's important because that's how Mendel did a lot of his work with pea plants, which gave rise to what we know today. So up until this point, we've been talking about traits and diploid and haploid and but how did scientists know what was responsible for heredity? What chemical component or molecule was responsible for carrying DNA? Today we know that it's DNA and RNA depending on the organism, but back then scientists didn't know. They had no idea. And so that was important for them to understand. Now, when you look at this diagram here, I want you to take a second to look at um, this diagram here from Griffin. And what you'll see here is a study that used a virulent strain of and a non-virulent strain. The non-virulent strain is the R strain. The virulent strain is the S strain. The R for rough and the S for smooth. Now, depending on what textbooks you read, you may get different variations of it, but ultimately, one of the major differences between the R and the S strain, which the strain is for pneumonia. It's a bacteria that specifically Streptococcus pneumonia. The R strain does not have a polysaccharide coat, which makes the outer portion smooth, which is what the S strain does. Now, some scientists believe that that S strain was composed of protein and so forth. Some scientists they believe that this is why for decades or for a long period of time that there was a debate between what was responsible for heredity or the transforming principle. Was it DNA or was it protein? Howbeit, they knew that the chromosomes consisted of protein and DNA, and so Griffith's initial studies helped provide that foundational understanding. And what did he do? He ran experiments on mice, which led to the discovery of bacterial transformation. Okay, and he laid the groundwork for scientists to understand more about genetics. What did he do? He injected the R strain into the mouse, and the mouse lived. He injected the S strain into the mouse, and the mouse died. So they said, well, let's heat kill the S strain because it was believed that polysaccharide coat, which may have been had the assumption of protein, was responsible for the virulence that was seen. So they applied heat to the S strain and then injected it into the mouse. The mouse lived. So surely it was thought that it was the smoothing or the polysaccharide coat, or then at that time maybe the protein, was responsible for heredity. And so what they did was they took the heat killed S strain, combined it with the live R strain, injected that into that combination into the mouse, and the mouse died. And so therefore it was believed that it was something that transformed from the S into the R, making it virulent and causing the mouse to die. But there was still work that had to be done to further understand what happened in this study. And so while something transformed, what was that chemical that was responsible for that? And this brings us to Avery, McLeod, and McCarty. Their study built off of Griffith's experiment and trying to help really understand, was it protein or DNA that was the carrier of genetic information. And so they did the same study, but in this case, they used the he killed S combined with the R. But before injecting into the mouse, they actually added enzymes that specifically targeted certain molecules. So proteases removed the protein, RNases degraded the RNA, DNases degraded the DNA, and they were able to see what happened. And in every case, the mouse died except for when the treatment was the solution, was treated with DNase, and this suggested that DNA was the transforming principle. So, of course, as scientists, we're very apprehensive and we question a lot. So this helped to provide another study that was informative and groundbreaking for our field in science, and that was from Alfred Hershey, and Margaret Chase. And what they did was the Hershey and Chase experiment. It's very, very historic, and you should know this experiment. They worked with bacteriophages to further show proof that DNA was the transforming principle and responsible for heredity. Just to briefly go over this, I won't go too much in detail, but because this will come back up again. But they provided solid proof that DNA, not protein, was responsible for heredity. Ultimately, what they did was they used a bacteriophage. Now, if you see in this diagram here, you have a bacteriophage. This is the capsid or the protein head. You have the tail fibers. Inside the protein or the capsid, okay, the capsid is made up of protein. Inside of that is where you have nucleic acid. Okay, in this case, our DNA would be housed there. And then, so ultimately, you're able to trace protein versus DNA. And so, what they did was they grew up the bacteriophages in a radioactive medium. One had S35 and the other had P32. Now, the reason why they used S35 and P32 radioisotopes is because sulfur is specifically found in protein. Examples of that would be like methionine, cysteine, and then you have phosphorus that is found in DNA. Remember, for those that may have you've had cell biology or even general bio, you know that DNA is composed of a sugar phosphate backbone. So growing the bacteria up in phages up in P32 means that all the DNA as it is copied and duplicated is going to incorporate radioactive phosphorus into the DNA. And so after they allowed the DNA to grow up in the radioactivity, they allowed the bacteriophages to infect bacteria cells. They checked for radioactivity, okay? And the protein radioactively labeled uh, study on the left, no radioactivity was found inside the cell. The only radioactivity found inside the cell was of DNA in the second experiment. But then you also had the study where they then apply centrifugation of the sample and then looked at radioactivity. And they found radioactivity in the supernatant in the experiment one, but in the other experiment, they found radioactivity in the pellet. And so this was a further validation that DNA is a genetic material based off of tracking the presence and the location of the radioactivity. So ultimately, understanding more about DNA really led to what we know today and gave rise to Watson and Crick and their ability to study and understand the structure of DNA. Now, we can't talk about the structure of DNA and the Nobel laureates Watson and Crick without talking about Rosalind Franklin. Rosalind Franklin's X-ray crystallography was actually the reason for Watson and Crick's ability to accurately decipher the structure of DNA. So Rosalind Franklin did an X-ray crystallography and left her lab book open on her desk, and Watson and Crick saw it. They were able to take that and piece together, combined with Chargaff's rule and other work that was done, the structure of DNA. And what they concluded was that DNA was a double helix that is anti-parallel, it's double-stranded, and it's made up of nucleotides. What are the nucleotides? A nucleotide is a monomer that is composed of a sugar, a phosphate, and a base. Okay, here is your sugar here. This is your phosphate, and this is your base. Okay, and these three together make up a nucleotide. Now, the four bases are adenine, thymine, cytosine, and guanine, and they engage in complementary base pairing. This also came from, in part, and built off of Chargaff's rule, which said there's a 1-to-1 ratio of bases. And so we know that adenine binds with thymine and guanine binds with cytosine. And this complementary base pairing across the helix allows for a double helix that is anti-parallel and double-stranded. Now, RNA is very similar to the structure of DNA. And actually, let me go back because I want to point something else out. When you look at the term DNA, DNA stands for deoxyribonucleic acid. Deoxy means without oxygen. And so when you look at the structure of DNA, you will see on the sugar here, while it doesn't show all the chemical molecules here, off of this second carbon here, off the sugar, there is no oxygen. And this is one of the differences between DNA and RNA. DNA does not have oxygen off of this second carbon. It does not have a hydroxyl group here, but it does have it on the third carbon in the sugar. RNA does. RNA stands for ribonucleic acid, and so off of that second carbon, there is a hydroxyl or OH group present. Another difference between DNA and RNA is that RNA is single-stranded. It is not double-stranded. Okay, it does not contain adenine. Instead, in place, it contains uracil. And so the base pairing is a little bit different because instead of A binding with T, it's U binding with A and G binding with C. I also want to point out, I guess I'll go back again to point out another difference here, and that is the number of hydrogen bonds that are present. In the backbone of DNA, you have a phosphodiester bond that forms the backbone of DNA, a sugar phosphate alternating backbone, so sugar phosphate, and then holding the two double strands together is hydrogen bonds that form between the bases that are complement to each other. So between adenine and thymine, you have two hydrogen bonds that form. In cytosine and guanine, you have three hydrogen bonds that form. And if you all remember from Gen Chem, or maybe you've learned about it in Organismal, that hydrogen bonds are not very strong bonds. They're weak, and so they can easily associate and dissociate. However, that does give DNA a little bit more complexity and roughness, if you want to say, than that of RNA. And that is why DNA is more stable than RNA is. RNA is not very stable. It actually will start to denature and degrade quickly, as opposed to DNA. DNA can be is a little more stable, and so ultimately you can find DNA at like a crime scene from 10 years, and it still be viable in a sense and be able to be sequenced, or while they can find that woolly mammoth that was frozen from thousands of years ago and still be able to isolate DNA from it and analyze it. So while they are very similar, they're still vastly different. So again, you all understanding and building off of the history over time of science has led us to also the understanding of the central dogma. Now, keep in mind, chapter 1 is a history, and it's just giving you an overview of genetics and some basic concepts. Most of these you all should have heard before, and so this should be a refresher for you. The central dogma is a theory that proposes the flow of genetic information. And this is where you have DNA that is transcribed into RNA and then RNA that is translated into protein. Now, there is accuracy to the central dogma, but there's also exceptions or flaws to it. And that is because it gives the perception that it is unidirectional. And we know that is not the case. There are some organisms whose nucleic acid is not DNA based, it's RNA based. And so what you end up having is a reverse transcription where RNA is reverted back into a form of DNA, which we call complementary DNA or cDNA. And so that lets us know right there that there's an exception to the central dogma and that it is not unidirectional. However, in general terms, right, while there are exceptions, the central dogma does help us understand how genetic information flows. And an easier way to understand it, or a more simplistic way, our genes, our DNA, which makes up our genes, carry the information in the form of nucleotides or bases that are then copied and transcribed into RNA. So there's a coding that changes, okay? Where remember, we no longer have T's, we have U. And then in order to go from RNA to protein, we engage in a transition or translation of bases to amino acids. And that is done through the triplet code, okay, or the genetic code, which we will talk about. But ultimately, this was an important and groundbreaking for what we know today. And this still is valid today, and it tends to be very universal. While there are exceptions, this is universally accepted amongst various organisms. Now, what is that genetic code? The genetic code says that for every 3 bases, it denotes 1 amino acid. And this is how we go from RNA to the production or the synthesis of protein. The machinery, enzymes, and other proteins scan the RNA, and every 3 bases corresponds to a specific amino acid. For example, as you see in the genetic code or this genetic table that is made up of 64 codons, UUU and the RNA would denote the amino acid or code for the amino acid phenylalanine. UUA would code for the amino acid leucine. What you should also notice here, and we'll cover this again when we go into translation, but the genetic code, there's only 20 amino acids, but there's 64 codons. Of the 64, 61 code for amino acids. The other three code for, or don't code for any amino acid. They actually stimulate a stop or termination response in translation. But what this ultimately says is that there's a level of degeneracy to the genetic code because there's more than one way to get the same amino acid. Focusing here at proline, there are 4 different codons that can produce the same amino acid proline, or serine, or threonine, or alanine. So there's more than one way to get the same amino acid, and this provides that complexity that they were trying to understand earlier in science. So what are proteins? Proteins are an essential part of what we do and who we are. Proteins make up our hair, they make up our nails, they are responsible for the hormones and the emotions we feel. They are responsible for even the food that we consume, right? Think about digestion. You have mechanical digestion, which is the use of your teeth to help break down your food. But then you also have enzymes that engage in that process to help with the breakdown and the digestion of your food, or the enzymes that are needed, or hormones that are needed to send signals to your brain of satiety when you're full, or the feeling of happy or sad. So proteins are very integral to life as we know it. They are made up of amino acids, and their basic form is called a polypeptide chain. And that is because it's a linear chain of amino acids that are held together by a peptide bond. So in this diagram here, you see, as it zooms in, is these small little circles, which are just representative of amino acids, are actually the different amino acids: phenylalanine, leucine, serine, cysteine. There are a total of 20 amino acids, but they're all different. They all have different chemical formulas, and also, as a result, respond and act differently when they are connected. And so you can have different combinations of these amino acids that form proteins that can either be enzymatic or non-enzymatic, but they all have an important function that are essential. And what you'll see is any abnormalities to these proteins can result in human diseases or disorders. And we can also see abnormalities in agriculture as well. One prime example in genetics is sickle cell anemia. Sickle cell anemia is derived from a protein abnormality in the beta chain of hemoglobin, and it is caused from a mutant form of that beta chain that makes up hemoglobin, and it prevents the protein from effectively carrying oxygen throughout the body. Well, when oxygen cannot be disseminated throughout the body, the different organs, you can have organ failure, you can have muscle spasms and pain. And so the experiences that a patient with sickle cell experiences can be vast. But what's interesting is this all starts from a single change in a base, a nucleotide, as you see here. And the normal, and here again, you'll see here is the flow of the central dogma, or the flow of genetic information. On the DNA level, the 6th position in the sequence for, or the amino acid sequence for the beta globin gene, you have CTC. CTC, when transcribed into mRNA, is GAG, and GAG, according to the genetic code, produces glutamic acid. Well, what's the problem with glutamic acid, or with this mutation, is that in the mutant form, that T is changed to an A. And when you change that second position, it produces a different mRNA. Well, changing the mRNA, as we've just seen, produces a different amino acid. In that case, you have valine. Why would that be important? Glutamic acid and valine are 2 different amino acids structurally, and so they behave differently, and it causes for the sickling in the red blood cells that you see. And the sickling of those red blood cells causes it to become stiff and sticky, and they aggregate in the vasculature and cause pain, and they can actually lyse or activate hemolysis. And so you have oftentimes patients with sickle cell need blood transfusions quite frequently. They engage in extreme pain. And so this is just from one change on the DNA level that is transcribed and translated into an abnormal protein. So how has the advancement of science and our understanding of even that concept led to what we know today in modern day medicine and technology? Our understanding of, for example, understanding of proteins led us to our understanding of restriction enzymes. They were first discovered in around the 70s with bacteria. And they found that these restricted enzymes can actually cleave at known or specific sites, regardless of the organism. They have a specific site where they will cleave. And so they were able to use that information to um, in various organisms. And they use, for example, these vectors. Vectors are these vehicles that carry DNA fragments, and they can form recombinant DNA. They are great for cloning and making genomic libraries. And so, utilizing this, you can undergo transformation and then screen for a specific bacteria for a set trait, and then take that and put it into a various organism for breeding or for a change in a different phenotype that you're interested in. This was found in Dolly. Dolly was a Finn Dorset sheep that was cloned from genetic material of an adult mammary cell. And next to Dolly, you see her firstborn lamb, which is Bonnie. Now, this was important and it was very groundbreaking because this was the first time that they were able to actually clone. And not only that, the clone was able to produce offspring. And so this was extremely important and groundbreaking in the field of biotechnology and genetics. So, what has been the continual impact of biotechnology in science and medicine as we know today? Biotechnology is now used in healthcare. It's used in supermarket products like the foods and the fruits that you consume that may give them a longer lifespan. It causes or it slows down the process of um, ripening. It's used in healthcare for the production of medications that are needed. It allows for the widespread distribution of vegetation and also using the court system for determining paternity. The Green Revolution was an important stage in history because, as you see in this diagram here, Norman Borlaug, he was the leader of the Green Revolution, and he was awarded the Nobel Prize in 1970 for his ability to produce a high-yielding rice plant, and was influential in expanding food and agriculture throughout the world, especially in places where it was food deserts or they were not able to get certain foods or missing nutrients. And so while sometimes society gives cloning and GMOs a bad rep, we know as scientists that there have been a widespread benefit to that because it has provided food in places that normally wouldn't get it. It has helped reduce food hunger, the lack of nutrients throughout the world, but also the ability to just increase the shelf life of certain vegetation long enough to distribute it around the world and go from one country to another. And so this has been extremely instrumental in what we know today, even when it comes to water retention and water reduction. This has also been a benefit in many different diseases and treatment. For example, most of us know someone that suffers from diabetes and takes Humulin or a form of human synthetic insulin. Well, where did that derive from? That derived from the use of biotechnology and what we just saw, cloning. Scientists for a long time were struggling with how to expand access to insulin at an affordable rate without triggering immune response or considering the patient's health. Before this technology came about, many patients were getting bovine insulin, so insulin from beef, and they were having immune reactions to it. It was expensive, it was difficult to obtain, and so there were so many issues with using that form of insulin. Well, scientists said, well, we know that bacteria can grow in a matter of hours, and so we can use cloning to force these bacteria to express the human form of insulin. Take the human form of insulin that is grown in these large tanks that you see here, purify the insulin from the bacteria that are growing there at a vast rate, purify and then bottle it for patients. And that is how we have modern day insulin today, or the use of anti-malarial drugs, antibiotics that are produced, or even now that we're seeing this advancement in personalized medicine, specifically in genomics and proteomics. So biotechnology and genetics and medicine are aligned with one another and are growing at a rapid rate. And so I want you all to have a greater appreciation for it by being aware of its benefits and how it actually does more good than the proposed harm that is often displayed. These are just some of the genes whose mutant forms cause human diseases and are currently being tested to find ways to cure or to counteract that disorder, whether we're talking about breast cancer, neurofibromatosis, which we'll talk about in an upcoming chapter 4, I believe. We talk about epistasis and we talk about penetrance and expressivity. You'll also see it in sickle cell anemia. You'll see PKU, Alzheimer's disease, Tay-Sachs disease. There's a list of various genetic diseases that are formed due to mutations. Genomics, proteomics, and bioinformatics are a new field, and they are expanding and doing wonders in what we know today in the field. The Human Genome Project created which was a $1 million project, millions of dollars, that was funded using taxpayers' dollars, but it was so instrumental. You can actually pull this up on the NCBI website to look at almost any gene sequence in the human genome, and this also corresponds, to many other organisms. But it created new disciplines. So now you have the discipline of genomics, which studies the structure, function, and evolution of genes and genomes, or proteomics, which identifies various proteins in a cell and how they can be altered based off of various conditions and how they interact with one another. Then you have the field of bioinformatics, which is more computational, but it uses software and computational methods to look at DNA, RNA, and protein data. All of those factors are important, and especially the Human Genome Project, because it allowed us to look at the etiology of certain genes and how they have altered between one organism or species and another. But also, it allows us to use various organisms for as model systems to study genetics before going into humans. For example, zebrafish. Zebrafish are one of the models that are used to study various traits, and that can correspond back into humans. For example, the golden mutant, which caused a change in the pigmentation in zebrafish, and it's a mutation that is caused by a gene that controls the amount of melanin that deposits within melanosomes. Well, this is seen in a similar gene in humans, and that's called the SLC24A5 gene. And this gene encodes a protein that has the same function in human cells. And so that's why you see this difference in pigmentation of these 3 different individuals here. But this is synonymous to what was seen in zebrafish. And so this is important because we're able to use other model systems that are considered lower species, no offense or intended, but to find an alignment that functions similar in humans. Because genes influence our lives. It determines weight, height, trait, hair color, pigmentation. They even control part of our personality. And we can also look at how environmental factors and external forces affect those genes and how they function. That's a term that has been in existence for a while, nature versus nurture. Is it nature that contributes to various traits or disorders or behavior, or is it nurture? At the end of the day, genes are fundamental to who we are and what we do. There are various modern approaches to understanding gene function, whether you're talking about classical or forward genetics, that's identifying genes that cause phenotypes, or reverse genetics, where a specific DNA sequence is known, but its function is not fully understood. And so there are reverse genetics to understand that function and tie it back to genetic sequences, or through gene silencing, RNAi, where you knock out a gene and you look at the function or the non-functional role that occurs when that gene is no longer present. This is something that I do in my lab, and we use different measures to do gene knockouts or RNA interference, whether it's through CRISPR or through other siRNA or shRNA, small hairpin RNA, to cause the silencing of various genes. Regardless, genetic studies rely on the use of model organisms. And so the question is, what determines whether an organism is a viable organism? Well, there's certain factors that have to be, or criteria that have to be met. It has to be easy to grow. It has to have a short life cycle, because if it takes 20 or 30 years just to see offspring, then it's probably not the best model to use. But not only that, it can't produce just one offspring, because then you can't really study the effects if you're only looking at one offspring produced. So that model system has to be able to produce many offspring, whether we're talking about flies that can produce hundreds of thousands of larvae that result in flies, or you're talking about E. coli, which can produce millions of offspring, or mice, which can produce dozens of offspring at one time. Um, Ultimately, we have to be able to rear this model organism in an environment and be able to control its lifespan and treatment. These are just some examples of organisms that can be used and their offspring to study various diseases, whether we're talking about E. coli and colon cancer and other cancers. Yeast is another one that was used. Drosophila, C. elegans, looking at diabetes, or mice looking at Ellen syndrome, cystic fibrosis, and various other diseases. We can use these different model systems combined with recombinant DNA technology to transfer genes between species and see how they respond, and see if the phenotype that is expected is altered when the genotype is altered or changed. And this is important. Or you can have the use of T phages, bacteria, to help study transfer of genes, infection. The, the amount of work that can be done is vast. However, we are living in the age of genetics, and genetics is just going to continue to grow. But we have to give credit to the father of genetics, Gregor Mendel, because his work that he did in the 1800s has set the stage and laid the foundation for what we know today. And that is, genes are passed on from one generation to the next, and being able to understand how they're passed on and being able to use modern day biotechnology and science to manipulate those genes allows us to develop cures for various disorders and diseases that humans are plagued with, sometimes self-inflicted. Howbeit, being able to modify it and develop treatment and drug therapy for it is essential to life as we know it. This shows you just how science works. And so sometimes when you are exposed to a new drug or a new form of technology, don't assume that it just happened overnight. Science is always building off of each other, and we owe what we know today to the forefathers of genetics and science, because they have helped build this whole postulates of science and theory that help us to what we have today. So what is the future of genetics, right? There's going to be a great advancement in personalized medicine, or the way we do prenatal testing, being able to own genes, or just having access to safe therapy, gene therapy, that may prevent the passing on of certain traits, which may also eradicate certain disorders. Now, keep in mind, there are going to be things that we talk about that have ethical um, questions, that raise ethical flags and awareness, but it's safe to talk about that. And it's a safe place to talk about it in the classroom, because where else should you talk about things like this? So I hope that you all enjoyed and learned something from chapter one. While it is filled with a lot of history and a lot of prerequisite information that you should be aware of, I hope that it helped bring things together, and it told a story that allowed you to follow the lifespan of science as we know.